Okay, thank you, Professor Tominaga. Thank you to those involved in organising this meeting. It's been fantastic so far. And also thank you to all the sponsors involved. My talk today is looking at new insights into TRPV4 as an integrator of cellular signalling. The talk will take the following organisations. So first I'll give an overview of G-protein coupled receptor, or GPCR, TRP channel interactions. I'll then present original work looking at histamine receptor TRPV4 coupling in lymphatic endothelial cells. And then, if time permits, I will then present some work looking at a multi-omics analysis of TRPV4 macrophages and the consequences of TRPV4 deletion on proteins and lipids. So it's well established that G-protein coupled receptors can interact with TRP channels and that TRP channels drive many pathways in GPCR signaling. The classic example is the Drosophila TRP channel, which promotes signaling by the G-protein coupled receptor rhodopsin, which is GQ coupled in response to light stimulation. And the pathway involved is summarized in the figure on the right. It is now known that there are many examples where GPCR and TRP channel interactions have important physiological and also pathophysiological roles. Regarding my sort of research interest in nociception and neurogenic inflammation, GPCRs are known to sensitize TRP channels, which leads to increased signaling from nociceptive neurons, and also produce endogenous activators, for example, activators of TRPV1. Sensitization of TRP channels by GPCRs also leads to enhanced neuropeptide release. This can lead to neurogenic inflammation related to barrier dysfunction and edema due to increased plasma extravasation, as well as altered lymphatic drainage. Importantly, GPCRs are ubiquitously expressed. They are probably the most targeted class of proteins with respect to clinically approved drugs. Approximately 40% of all drugs target some aspect of GPCR signaling. It is established that a broad range of GPCRs can signal via TRP channels. The mechanisms involved are summarized very briefly here. This includes changes to membrane fluidity, altered cell surface expression or even internalization of the channels, production of endogenous activators, for example, arachidonic acid derivatives, protein-protein interactions, for example, interactions with G proteins, or sequestration with beta-arrestins as part of a scaffolding process. And TRP channels are also modified by post-translational changes, including phosphorylation, which generally enhances signaling, and ubiquitination, which can lead to delivery of TRP channels to lysosomes of degradation, effectively terminating their signaling. Providing two examples here. These are related to TRPV, tripvanilloid interaction with the protease activator receptor 2. Shown on the left is one of the earlier studies. This is Dye et al. 2004, where they looked at sensitization of TRPV1 in nociceptive neurons. And they've activated PAR2. This is the capsaicin response prior to PAR2 activation. And then following activation of PAR2 with synthetic ligands or proteases, you can see a marked increase in the capsaicin-induced current. And this is mediated through protein kinase C activity, shown here using inhibitors. Our own work has defined the importance of TRPV4 for the sustained plateau phase of signaling. So this is a calcium response to PAR2 activation. And this part here, the plateau phase, is driven by TRPV4 activation, as shown here. So this is with TRPV4, and these are HEC cells without TRPV4. This effect is driven by the production of endogenous lipid activators of TRPV4, as well as phosphorylation by a tyrosine kinase. So TRPs can be sensitized by GPCRs, and they can also couple to drive GPCR-dependent signaling. This diagram here sort of summarizes the sort of current state of our knowledge regarding how different classes of GPCRs may interact with TRP channels to influence their activity. Generally speaking, GI-coupled receptors are considered to be inhibitory in terms of TRP activity, whereas GS will couple through PKA activation, and GQ-coupled receptors will activate TRP channels through either lipid or kinase-mediated pathways, generally speaking. So my main research interest is in digestive diseases and the gastrointestinal tract. And as part of this research, we were interested in defining where TRPV4 is expressed within the gut wall, given that it is responsive to mechanical stimulation, although that has been questioned recently. The most prominent site of expression was within these initial lymphatic capillaries. So these are blind-ended capillaries which only contain endothelial cells. So there's no smooth muscle coverage, and there are no valves present within the structure here. This is calcium imaging shown here within a whole mount preparation of the gut wall, and we've shown this in the mouse colon, monkey, as well as human colon. So this is consistent across species. As you can see, there's, in response to the synthetic agonist GSK101, there's a very marked increase in both cytosolic and nuclear calcium within these endothelial cells. Shown here is another preparation. This is a lymphatic vessel over here. This is a post-stained image over here showing live 1 positive lymphatic vessel and a CD31 positive capillary. And on the left, you can see the lymphatic response in black and the response by the blood vessel in orange. These effects are effectively abolished in a TRPV4 knockout animal, consistent with a TRPV4 dependence. And if we do faster imaging, you can see that the response by blood vessels is oscillatory, this is calcium, and then the responses by lymphatic vessels are more sustained. We decided to examine this in more detail, looking at primary human lymphatic endothelial cells. These are dermal lymphatic cells, and this is in a culture. And you can see the calcium response. Again, a very prominent nuclear calcium. This is concentration dependent. This is activation with GSK101. And the effect is blocked by TRPV4 inhibition. So we wanted to examine GPCR TRPV4 coupling in these cells. Surprisingly, little is known about which GPCRs are expressed by lymphatic endothelial cells. The most prominent ones in the literature will be the adrenomedallin receptor, as well as the S1P receptor. We decided to look at histamine receptors instead. Histamine 4 receptor came up in our single-cell RNA-seq data, as well as information from the literature. So lymphatic vessels are surrounded by mast cells, which produce histamine, and these are required for acute inflammatory changes to the lymphatic vessels, as well as trafficking of immune cells to draining lymph nodes. There's also some evidence that LEX, lymphatic endothelial cells, express histidine decarboxylase and may also contain histamine. They also, histamine is also one of the mediators of shear stress-induced relaxation of lymphatic vessels. And this is mediated through H1 and H2 receptors. This, evidence from the literature shows that histamine receptors can signal via TRPV4 in native systems. Shown here is an example from dorsal root ganglion neurons. These, that's the GSK101 response here, and that's following application of histamine. So you can see there's a marked increase in the responsiveness. This has also been shown by electrophysiology in this paper here. And this is related to visceral pain in irritable bowel syndrome, as well as in acute colitis. TRPV4 may also drive the calcium responses to histamine receptor activation. This paper here looked in primary keratinocytes and showed that signaling by the histamine 1, 3, and 4 receptors are TRPV4 dependent. And that's related to pruritus in response to histamine. And then just to add another example, this paper here, recent paper, showed that plasma extravasation in the mouse ear in response to histamine was suppressed in TRPV4 knockout mice. So these papers basically indicate that histamine receptor activation can sensitize TRPV4 and that TRPV4 may promote histamine receptor signaling. So we examined the expression of histamine receptors in lymphatic endothelial cells using a pharmacological approach. And these are calcium responses. It's concentration-dependent responses to histamine. And shown here on the right, we can see that histamine response is effectively rightward shifted in the presence of an H1 blocker, as well as attenuated in the presence of a well-characterized histamine 4 receptor blocker. So we then examined which histamine receptors may interact with TRPV4 in these cells. Shown here is the response to histamine. So this activates all receptors, shown here. And the response is suppressed in the absence of calcium, so nominally calcium-free media, as well as in the presence of a TRPV4 antagonist. If we examine the histamine 4 receptor only, so note that histamine 4 is a GI-coupled receptor, we can see that we get a calcium response to 4-methylhistamine, which is currently the most selective agonist of this receptor. And this is effectively blocked by the TRPV4 antagonist, by the removal of extracellular calcium, as well as by a histamine 4 receptor antagonist, consistent with histamine 4 driving calcium signals through the TRPV4 channel. We also looked at the sensitization of TRPV4 signaling. So these studies involve pre-stimulation of the cells with a histamine receptor agonist, followed by a concentration response curve to GSK101. The response to histamine GSK101 by itself is shown in black in cells pre-treated with histamine, so it's a leftward shift. And then in the case of cells pre-treated with the histamine 4 receptor agonist, you can see there's probably a 100-fold shift, leftward shift, so quite a remarkable sensitization of the TRPV4 channel. We then examined the potential mechanisms, and we decided to focus on the role of PLA2 based on information from the literature. So to do this, we used two well-established inhibitors of PLA2 forms. So this is soluble and cytosolic PLA2. And as you can see, compared to the response to 4-methyl histamine pre-treated cells, the response to GSK101 is rightward shifted, so it's blocked. So this is a current working model. We have PLA2-dependent production of endogenous TRPV4 activators. There is some role for PKA. There is no role for PKA because it's a GI-coupled receptor, so it blocks adenylate cyclase activity. We also studied this interaction using pertussis toxin, and there's a minor effect of pertussis toxin. So GI-O coupling, there is some evidence for that, but not a great deal. We then looked at the downstream effects. So right now we're sort of examining calcium response, but we wanted to know what the consequences of the signaling were. So we looked at junctions, adherence junctions in cultured lymphatic endothelial cells. So activation of TRPV4 led to the formation of these linear junctions and remodeling of the actin cytoskeleton. And this was effectively blocked by a TRPV4 antagonist. Histamine promoted discontinuous junction formation and remodeling of the actin cytoskeleton, which was reduced by TRPV4 activation. It's not as prominent as the response to GSK101, as would be expected. And 4-methylhistamine also had a minor role in these changes. Please note I have not quantified these images because it wasn't as simple as I thought it would be. Any ideas how to do that, please let me know. So we looked at the NFAT signaling, NFAT-C1 and 2. This is a transcription factor that is activated by calcium and calcineurin, and it detects oscillations in intracellular calcium. It's important in the regulation of lymphatic development, as well as in maturation and maintenance of vessels. So we examined this by microscopy. Shown here is a positive control ionomycin, so just to allow calcium influx. In the vehicle, this DMSO. So you can see there's a marked increase in nuclear calcium, nuclear NFAT-C1 2 labeling. Similar effects were observed when cells were activated with GSK101, the TRPV4 activator, and these were TRPV4 dependent, and also with treatment with histamine and 4-methylhistamine. So these effects are all consistent with NFAT-C1 translocation to the nucleus in response to histamine V4 activation. And these data are summarized on the right here. And so these findings have implications for the transcriptional control in LEX. We're particularly interested in looking at sort of chemokine and cytokine production. And to do this, we looked at using a bioplex assay. So we measured more than 20 different chemokines and cytokines. And the idea was that we would identify maybe some pathways that would lead to promote chemotaxis into lymphatic vessels and then delivery of cells to draining lymph nodes. So shown here on the left, so generally no effect on MIP1 alpha. This is after 24 hours stimulation. PWU, which is our positive control, did upregulate release of MIP1 alpha. MIP1 beta was activated in a TRPV4 dependent manner. This is GSK 101, and also a histamine TRPV4 dependent manner. And then CCL5 is shown on the right. This is increased with PDBU and GSK 101, but not by ionomycin. So this is TRPV4 dependent, but there is no effect of ionomycin suggests this is not due simply to elevated intracellular calcium. So I guess the question is TRPV4 actually important in lymphatic vessels? TRPV4 is activated by shear stress. That's one of the activation mechanisms. But as far as I can tell in the literature, the shear stress, fluid flow and shear stress within lymphatic vessels is so low that it's unlikely to activate TRPV4, which is activated around 4 dynes per centimeter squared. We decided to look at interaction between piezo1 and TRPV4 due to the role of piezo1 in lymphatic development. So it's critical for formation of valves. This is based on knockout animals, but also in human mutations. And also the increased piezo1 expression or activation can increase valve development. And there is a set point theory which predicts that there's a requirement for several mechanosensors with differing sensitivities for the cellular function. We decided to work on this based on this study here by Roger Little's group, which showed that in pancreatic acinar cells, TRPV4 and piezo1 can interact to drive pancreatic changes, so pancreatitis. Based on this model here, piezo1 will promote the release of endogenous arachidonic acid derivatives which activate TRPV4. We examined this in the mouse colon, so these are calcium imaging of tissue. We decided to use Yoda1 as the activator, given the challenges with manipulating the intestine. And we showed that TRPV4 knockout animals had suppressed responses to Yoda1, consistent with interaction between piezo1 and TRPV4. We also performed these same studies in cultured human primary LEX. Shown on the top is TRPV4 responses completely blocked by TRPV4 block, and then Yoda1 responses, which is suppressed by the TRPV4 antagonist HC067. As predicted, we showed that TRPV4 has no role in the responsiveness of cultured lymphatic endothelial cells to shear stress. These are cells grown within a microfluidic device and included with or without a TRPV4 antagonist, and this had no effect on the positioning of the nucleus, the organization of the cells, their volume, or any other of the key features. And we also examined the lymphatic development in the ears of TRPV4 null mice. In this case, we stained with CD31, smooth muscle actin, and live1, and there was no difference in the formation of valves or the organization of lymphatic vessels in these mice, consistent with perhaps a role for TRPV4 in facilitating piezo1 signaling, but not a requirement for TRPV4 for the function of lymphatics or their development. So to summarize the key findings, we showed that there is likely to be piezo1 TRPV4 interaction in lymphatic endothelial cells. We showed that the histamine 1 and 4 receptors can interact to activate TRPV4 or sensitize this activity. And we showed that downstream of TRPV4, signaling, there was a translocation of the transcription factor NFAT, which resulted in alterations to the cytoskeleton and also to the release of cytokines. So these findings have implications for fluid balance and immune function. We have not examined this in vivo. For example, with respect to lymphatic drainage, chemotaxis, changes in inflammation, even perhaps responses to food allergy, things like that. In terms of future considerations, one of the major advances, certainly in the GPCR world, is the idea that a receptor can continue to signal from different parts of the cell following internalization. We are actively examining whether TRPV4 may also signal from different cellular locations. We've previously shown in unpublished or unpublishable findings that TRPV4 is constitutively internalized and is recycled, and we also have evidence that TRPV4 can promote signaling within mitochondria and the nucleus. One limitation of this work is a singular focus on calcium signaling and the use of pharmacological activators as opposed to endogenous sort of means to activate the channel. We are also looking at other downstream pathways, for example, kinase signaling, or the formation of complexes using approaches such as BRET or FRET. That's the next step, I guess. I think I've run out of time, so I'm going to skip over this. If you want to talk to me about sort of multi-omic analysis of macrophages and changes associated with knocking out TRPV4, please come and talk to me. I'm happy to talk. This includes lipid biosynthesis as well as proteins. So I will finish my talk with acknowledgements. This work is driven largely with my long-term collaborator, Nick Veldhuis. We run a group together. Nathan Nguyen has done a lot of the work, as have Dr. Jeffrey Rittermal and Scott Pang, who recently passed his PhD, which was a big milestone. Funding was from these sources here, more generally. If you're interested in the sort of work that we do, please visit our lab website. The QR code is up there. Thank you.
Thank you, Daniel. Are there any questions?
Thanks very much, Daniel. Are there any inherited disorders, or if you get inherited mutations in TRPV4, does it affect lymphatic, visceral pain, etc.?
Yes, we're actively working on that. So they are mainly related to like arthropathies and sort of motor neuron disorders. I don't know anything about lymphatic drainage, but we're looking at more in the context of, what are those cells called? Osteoclasts, those sort of cells. So related to lymphatics, I don't know. Yeah.
Very nice talk, thank you. Maybe I missed something. You showed that if you use an agonist for TRPV4, you can induce NFAT, nuclear translocation, and cytokine production. But so that means that TRPV4 can do that, but does it actually do that? If you knock it out, do you see that this doesn't work?
So I guess the best way to answer that would be related to the macrophage work I didn't show. So we've shown that TRPV4 is involved in, for example, interleukin-1 signaling. A bit hard to measure, but we've shown that that is a driver, and that's independent of other activators. So it's simply knocking it out can influence type 1 interferon signaling. So perhaps that's an example where it may be the case. Yeah.
Okay. Is there another question? So otherwise, I would like to ask you for the macrophage stuff, if you could just show that slide. Unless there's another question.
I can certainly do that, if that's okay with everyone. Okay, so what I haven't shown is that TRPV4 is prominently expressed by different macrophage subtypes. We decided to look at this in bone marrow-derived MACs. We used wild type versus V4 knockout mice. And these were either unstimulated, stimulated with GSK101, or stimulated with LPS. We then looked at metabolomic and lipidomic analysis and proteomic analysis. Simply knocking out TRPV4 led to quite marked changes in the proteome, as you can see from the heat map. Over here, so it's vehicle versus vehicle. It's a little bit annoying, I know. That's wild type versus V4 knockout. And perhaps it's a bit easier to see. So this is vehicle versus vehicle. You can see the upregulation of proteins and downregulation of proteins, and this is in response to LPS stimulation. We've validated some of the key pathways and key hits as well. I just don't have these data on me. And we've also have evidence that TRPV4 may influence lipid biosynthesis under basal conditions. So on the left is the cholesterol biosynthesis pathway. This is downregulation. So upregulation and then downregulation on the right here in the ceramide sphingolipid biosynthesis pathway. Again, a whole project that I couldn't present, but I'm happy to talk about it. Yeah.
Thank you. Are there any questions? Okay, Daniel, thank you very much.
Thank you.
I'd like to thank for speaking.